Somewhere between 5 and 7 million years ago, an ape lineage split in two. One branch became chimpanzees, the other became us. And for a long time, the story of what separated those two paths focused on the obvious things: walking upright, growing brains, making tools, developing language. Humans and chimpanzees share nearly all of their DNA, and yet within the tiny fraction that separates us are the changes that produced language, technology, and the most energetically expensive brain ever evolved by any animal on Earth. The human brain burns through roughly 20% of the body's entire energy supply just sitting still. By normal evolutionary logic, an organ that costly should never have been kept. Evolution does not tolerate expensive mistakes. So why did it stay? What pressure was powerful enough to justify that cost and to keep driving it upward, generation after generation, for millions of years? The answer most people have never heard points to something that was already happening long before the first stone tool was ever held in the first human hand. A pressure so constant, so relentless, and so invisible that it rarely appears in the version of human evolution most people are taught. It was not a predator. It was not a drought. It was not a competitor from another species. It was something that could not even be seen. And it may have done more to shape what we became than almost anything else in our evolutionary history. The evidence is not buried in the fossil record. It is written into something far more intimate, running silently inside you right now, in parts of your biology you have never once thought to question. Tonight, we trace that conflict, what it was, what it demanded of our ancestors, and what it quietly built inside us in return. If you enjoy stories like this, subscribe and leave a like. There is a lot more of this coming. Now settle in. This one changes how you think about what you are. Long before human beings gave names to nature, life was already locked inside a continuous negotiation with survival. Nothing living arrived in the world complete. Every feather, every tooth, every nerve, every instinct existed only because something before it failed, often enough for a different version to persist. Evolution does not move toward perfection in the way people often imagine. There is no summit waiting ahead, no final form toward which life steadily climbs. There is only pressure arriving endlessly from every direction, shaping whatever survives long enough to reproduce. A small variation is enough to alter the course of a lineage. A slightly thicker skin, a sharper memory, a body chemistry less vulnerable to infection. If that variation helps an organism remain alive long enough to produce offspring, the trait spreads quietly into the next generation. If it does not help, it slowly disappears into extinction. Natural selection is not guided by intention or wisdom. It is simply the uneven survival of differences, repeated across immense stretches of time until entire species are transformed by accumulated outcomes. Some of those pressures emerge from the physical world itself. Ice ages reshape forests into grasslands. Rivers alter their paths. Droughts erase food supplies. Mountains separate populations for thousands of years. These are abiotic pressures, forces created by non living parts of the environment. They can be severe, but they move with a certain slowness, allowing species time to drift and adapt. The deeper instability comes from living things. Predators improve their hunting, prey improve their escape, parasites refine their invasion strategies, hosts strengthen their defenses. Every adaptation on one side becomes a new pressure on the other. Unlike climate or geography, living opponents do not remain still while they are being adapted to, they respond. This creates biotic pressure, a form of evolution that accelerates itself through endless feedback. Life becomes both the sculptor and the stone. In 1973, evolutionary biologist Lee Van Valen examined the fossil record while working at the University of Chicago. He noticed something strange hiding beneath millions of years of extinction data. Species that had survived for vast stretches of time were not becoming safer from extinction. Ancient species remained just as vulnerable as younger ones. Survival did not accumulate into security. Every lineage, no matter how old, remained trapped in constant competition with the changing world around it. To describe this pattern, Van Valen borrowed an image from Lewis Carroll's Through the Looking Glass. In the story, the Red Queen tells Alice that running as fast as possible is necessary simply to remain in the same place. Van Valen realized the same principle governed life itself. Species evolve not because they are steadily advancing toward superiority, but because standing still means falling behind organisms that continue changing. This is the foundation beneath the human story. Three immense biological arms races shaped the species that would eventually become modern humans. The first unfolded between hosts and the pathogens that fed upon them. The second emerged within human social groups, where intelligence became entangled with rivalry, cooperation, deception, and status. The third entered through attraction itself, where mate choice quietly influenced which minds, bodies, and behaviors would continue into future generations. None of these races ever truly ended. Life simply kept running beside life, adjusting to pressures created by other living things that were running just as hard. Far beneath the visible world, beneath forests and oceans and animal migrations, life has always existed inside another landscape entirely, one made of intrusion, resistance, and survival at microscopic scale. Long before mammals appeared, before dinosaurs crossed ancient floodplains, before trees rooted themselves across continents, living cells were already under attack from smaller living things seeking a way inside. The oldest war on Earth began more than 500 million years ago, possibly earlier, in seas populated mostly by soft bodied organisms drifting through dim prehistoric water. Even then, survival was unstable. The moment one organism discovered a way to feed from another without immediately killing it, evolution found a strategy powerful enough to spread across nearly every branch of life. Parasitism became not a rarity, but one of the dominant lifestyles on the planet. Today, more than half of known species are parasitic during some part of their existence. Tiny worms inhabit intestines, fungi invade insects from within, protozoa travel through blood, viruses enter cells and convert them into factories for their own replication. Some parasites are so dependent on hosts that they can no longer survive independently at all, their bodies reduced over millions of years into streamlined biological tools built almost entirely for invasion. Every multicellular organism that has ever lived inherited this pressure. The emergence of larger bodies created opportunity for exploitation. A single cell drifting alone in ancient water offered little territory to occupy, but clusters of cooperating cells created shelter, nutrients, warmth, and stability. Life became richer and therefore more vulnerable. Some evolutionary biologists suspect that multicellularity itself may have accelerated partly because groups of cells offered better protection than isolated ones. Organisms became fortresses because the world around them demanded walls. At the center of this conflict lies a simple repeating mechanism. A pathogen evolves a molecular structure capable of attaching to a host cell. If the structure fits, entry becomes possible. The host lineage then changes the shape of the receptor, subtly altering the lock. Over generations, The pathogen evolves a modified key. Then the host changes again. The cycle continues endlessly, not through intention, but through accumulation of survivors. Viruses brought this process to extraordinary speed. Influenza, for example, can complete a new generation in roughly 6 hours. Inside a single infected cell, viral machinery may produce 100,000 new particles before the cell ruptures. Each replication introduces opportunities for mutation. Most changes vanish immediately, but occasionally one grants a slight advantage. Faster attachment, better evasion, improved transmission. Evolution that might require thousands of years in large animals can occur in viruses between one sunrise and the next. Against this pace, the human lineage appears almost motionless. Large primates reproduce slowly. Human generations unfold across roughly 25 years. A virus may experience millions of generations, while a single human family line advances by only a few descendants. The imbalance is immense, almost difficult to fully picture. Biological time moves differently for microscopic organisms, and yet multicellular life persisted. Ancient oceans did not dissolve entirely into infection. Forests still formed, nervous systems emerged, brains expanded. Somewhere inside this imbalance, evolution found methods for slower creatures to survive against enemies that could transform themselves almost overnight. Inside the marrow of human bones, hidden far from sight or sensation, cells continuously assemble molecules that have never existed before in the history of life. Tiny fragments of genetic material are cut apart, rearranged, and stitched together into new combinations, producing recognition structures capable of binding to shapes the body has not yet encountered. This process begins early in development and continues quietly for decades. The human immune system operates through two connected layers of defense. The older branch, called innate immunity, reacts rapidly and broadly. It identifies common signs of danger, fragments of bacterial walls, damaged tissue, foreign genetic material. Its response is immediate, releasing inflammation, fever, and chemical distress signals into surrounding tissue. This system is ancient, carrying traces of defensive strategies that emerged long before mammals existed. The second branch altered the balance of the conflict between hosts and pathogens in a more flexible way. Adaptive immunity does not merely recognize danger, it remembers it. Specialized white blood cells produce antibodies, proteins whose surfaces can physically attach to invading organisms with extraordinary precision. The remarkable detail is that the body does not build these antibodies in direct response to specific diseases. Instead, it generates enormous variation in advance, then preserves whichever forms prove effective. At any given moment, the adaptive immune system may be capable of producing around 10 billion distinct antibody shapes. Most will never encounter anything relevant. Some may bind weakly to harmless particles drifting through the bloodstream, but occasionally, among this immense molecular library, one antibody happens to fit a pathogen closely enough to interfere with its spread. The cells producing that antibody then multiply rapidly, filling the body with copies. What unfolds inside an infection resembles evolution compressed into the span of a single lifetime. Random variation appears first. Useful variants reproduce more successfully. Ineffective ones disappear. Every illness becomes a selective event occurring within living tissue. The body internalizes the same principles shaping species across geological time, creating temporary populations of competing immune cells and amplifying whichever performs best against the immediate threat. This extraordinary diversity emerges through a genetic process called V, D, and J recombination. The human genome contains approximately 3 billion base pairs and around 25,000 genes, far too few to directly encode billions of unique antibodies individually. Instead, immune cells shuffle modular gene segments into new arrangements each time an antibody producing cell matures. Limited raw material becomes an almost inexhaustible source of biological variety. Yet precision carries danger beside protection. A system capable of recognizing nearly anything risks eventually recognizing the body itself. Normally, immune cells that react strongly to healthy tissue are eliminated during development. Some escape this filtering process. Under certain conditions, the immune system begins targeting joints, nerves, intestines, or skin as though they were hostile organisms. Autoimmune diseases are not separate from the adaptive immune system's success. They are consequences woven into its design. A defense capable of infinite subtlety cannot remain perfectly restrained forever. The cost of building a biological structure flexible enough to confront rapidly evolving pathogens is the possibility of accidental self destruction. The immune system, therefore, becomes more than protection alone. It is one of the clearest physical records of the ancient evolutionary struggle still operating inside the human body, written not in stone or fossils, but in living cells endlessly adapting to survive. More than 2 million years ago, a subtle alteration appeared somewhere within the human lineage, likely among populations of Homo erectus moving through ancient Africa. It left no immediate mark on the skeleton. Fossils cannot show it directly, yet the mutation transformed something even more fundamental than bone. It changed the chemistry coding nearly every cell in the body. The outer surface of a cell is not smooth. It is covered in branching sugar molecules called glycans, intricate structures that help cells identify one another and regulate immune activity. To pathogens, these sugars are opportunities. Viruses, bacteria, and parasites often recognize specific molecular patterns on cell surfaces before invasion begins. A successful infection frequently starts with the correct molecular fit. Most mammals, including chimpanzees, carry a sugar molecule called Nu5GC on their cells. Early human ancestors once produced it as well. Then a mutation disrupted the gene responsible for manufacturing that molecule. Human cells gradually shifted toward displaying a closely related sugar called Nu5ACE instead. The chemical difference between the two is extremely small, yet its evolutionary consequences spread across the entire species. Evidence suggests that pathogens helped drive this transition. Certain infectious organisms had adapted to recognize new 5GC as a doorway into cells. Ancient relatives of malaria parasites are among the likely pressures. Individuals lacking new 5GC may have possessed a survival advantage during outbreaks, making the altered chemistry more common over successive generations. Once this shift spread through the population, the immune system itself began adapting to the new cellular landscape. A family of proteins known as siglix interacts directly with these surface sugars. Siglix function partly as molecular interpreters, allowing immune cells to distinguish between healthy tissue and potential danger by reading glycan patterns across cellular membranes. When new 5GC disappeared from human biology, the signaling environment surrounding Siglix changed dramatically. Comparative genetic studies show that Siglic genes in humans evolved with unusual speed after this transition. The genome preserves evidence of intense selective pressure concentrated around these proteins, far greater than what appears in many closely related primates. This wave of rapid cyclic evolution unfolded between roughly 2 million and 600,000 years ago. It became embedded within the genomes of Homo erectus, Neanderthals, and eventually modern humans. The consequences spread quietly through the body, reaching far beyond simple disease resistance. Several evolved cyclic variants are active in immune cells where they regulate inflammatory responses and pathogen recognition. Others appear in the placenta, helping manage the delicate immune balance required for pregnancy. Some are expressed in the gut, where the immune system negotiates continuously with vast microbial populations living inside the digestive tract. Certain cyclics are also present in the pancreas and throughout parts of the brain, suggesting that this ancient molecular rewrite influenced multiple biological systems simultaneously. The transformation was not merely defensive. Human cells adopted a new biochemical identity, and the immune system evolved alongside it. Pathogens adjusted in response, continuing the ancient cycle at microscopic scale. Every generation inherited slightly modified versions of the same molecular conversation. Even now, the legacy of that transition remains active beneath awareness. The surface chemistry of human cells still differs from that of our closest relatives. Immune proteins continue interpreting those signals in every organ and tissue. Hidden inside ordinary cellular function is the record of an evolutionary negotiation that reshaped the human lineage from the inside outward. For immense stretches of evolutionary time, certain regions of DNA remained almost perfectly stable. Across fish, reptiles, birds, and mammals, many sequences changed so little that scientists realized natural selection had been protecting them for hundreds of millions of years. Mutations still appeared, but most vanished quickly, filtered out before they could spread. These sequences persisted through deep geological eras with extraordinary precision. Then, within the human lineage, something unusual happened. Researchers, comparing the human genome to those of other mammals, discovered roughly 2,700 regions that broke this ancient pattern of stability. In species across the vertebrate tree, these stretches of DNA had remained highly conserved, yet in humans, they accumulated mutations rapidly and unexpectedly. Geneticists named them human accelerated regions, or HARs. The word accelerated does not mean chaotic. The changes followed a direction strong enough to leave a visible signature inside the genome. Mutations gathered in humans far faster than the neutral evolutionary rate would predict, while the same regions stayed relatively unchanged in chimpanzees and other mammals. Some new pressure had begun reshaping these sequences, specifically within human ancestors. What made the discovery even stranger was the location of most HARs. The majority are not found inside ordinary protein coding genes. Instead, they occupy regulatory regions, segments of DNA responsible for controlling when nearby genes become active, where they activate, and how strongly they operate during development. Genes themselves can be imagined as biological tools already shared across much of animal life. Regulatory regions determine how those tools are used. A slight shift in timing during embryonic growth can alter the structure of neural circuits. A change in activation strength can influence how densely neurons connect or how long certain cells continue developing before maturation. Many HARs appear to function as enhancers, molecular switches increasing the activity of nearby genes. Studies suggest that around 67% of tested HARs display enhancer behavior. Their activity becomes especially concentrated during fetal brain development, precisely when the architecture of the nervous system is being assembled. The pattern inside the genome is difficult to ignore. HARs cluster near genes involved in cortical expansion, neural migration, synapse formation, and cognitive processing. Evolution accelerated most intensely in regions associated with building and organizing the human brain. The genetic changes shaping humanity were often not the invention of entirely new genes, but alterations in how ancient genes were regulated. Positive selection appears to have driven these mutations forward. Beneficial variants spread through populations because they improved survival or reproduction under the pressures acting on early humans. The changes were not simply random drift accumulating over time. Something in the environment favored faster cognitive adaptation strongly enough for these accelerated regions to expand across the lineage. Exactly what forces created that pressure remains uncertain. Pathogens, increasing social complexity, communication demands, shifting climates, and competition within human groups may all have contributed simultaneously. Evolution rarely operates through a single cause. Different pressures often overlap, reinforcing one another across thousands of generations. What remains visible today is the genomic record itself. Sequences that stayed nearly frozen across vertebrate history suddenly entered a phase of rapid transformation during human evolution. Hidden among 3 billion base pairs are concentrated pockets of accelerated change, especially around the systems responsible for perception, memory, language, and thought. The human mind emerged not from one dramatic mutation, but from countless subtle adjustments accumulating inside the regulatory machinery, guiding how the brain develops itself. Long before language filled the air with stories, warnings, songs, and memory, the foundations for speech were already forming silently inside developing brains. Networks of neurons learned to coordinate breathing, tongue movement, facial muscles, hearing, timing, and fine motor control with extraordinary precision. Human speech appears effortless in ordinary life, yet beneath even a simple sentence lies one of the most complex biological systems evolution has assembled. Among the genes involved in this system, one became especially important to scientists studying the origins of communication. It is called FOXP2. Popular descriptions sometimes label it the language gene, though the phrase simplifies a far more intricate reality. FOXP2 does not contain language itself. Instead, the protein produced by the gene regulates the activity of many other genes involved in neural circuitry, vocal coordination, and aspects of speech and learning. Its evolutionary history is unusually ancient. Versions of FOXP2 exist across vertebrate life, including birds, reptiles, and mammals. Over roughly 450 million years, the core coding sequence changed very little. Natural selection preserved it with remarkable stability, suggesting that major alterations were often harmful enough to disappear quickly from populations. Then, within the human lineage, two amino acid changes appeared in the FOXP2 protein. The modifications are chemically subtle, yet they distinguish humans and our closest extinct relatives from chimpanzees. Neanderthals carried the same variants, meaning these changes emerged before the evolutionary split between their lineage and ours. The surrounding DNA reveals an even more striking pattern. Around the FOXP2 locus lie dense clusters of human accelerated regions, rapidly evolving regulatory sequences that influence when and where the gene becomes active during brain development. These regions do not rewrite the structure of the FOXP2 protein itself. Instead, they alter the timing, coordination, and intensity of its activity across developing neural tissue. This distinction is important because small regulatory shifts can reshape entire developmental pathways without requiring entirely new genes. The genomic territory surrounding FOXP2 became one of the fastest evolving non-coding regions in the human genome. Evolution concentrated pressure not only on vocal control, but on the broader neural architecture supporting communication. Research continues to show that these ancient changes remain biologically relevant today. A study published in 2025 followed 350 children through long term language testing and validated the findings across cohorts exceeding 30,000 individuals. Researchers found that the integrity of rapidly evolved genomic regions near FOXP, two binding sites, strongly predicted language performance in modern humans. Ancient evolutionary modifications still influence communication ability thousands of generations later. The consequences extended beyond speech mechanics alone. Once information could move reliably through language, human social life changed fundamentally. Knowledge no longer depended entirely on direct experience. Skills, alliances, warnings, rituals, and strategies could be preserved and transmitted across groups and generations with increasing accuracy. This created new evolutionary pressures within human societies. Individuals better at communication gained advantages in cooperation, teaching, persuasion, and social coordination. At the same time, listeners capable of detecting nuance, intention, and emotional meaning more effectively also benefited. Communication itself became part of the evolutionary competition shaping the species. The result was not simply a primate capable of producing complex sounds. It was a nervous system increasingly specialized for symbolic exchange and shared abstraction. The same evolutionary pressures shaping immunity and cognition also reached into the foundations of language, gradually constructing the biological framework through which human minds would eventually connect, remember, and imagine together. The human brain is usually framed as a triumph of intelligence, creativity, and social complexity. Yet another possibility emerged from evolutionary theory, quieter and far less comforting. Part of the brain's expansion may have been driven by the need to resist microscopic organisms capable of altering behavior itself. Parasites do not simply consume tissue. Many interfere directly with the nervous systems of their hosts. Certain fungi guide insects toward locations favorable for spore dispersal. Some parasites reduce fear responses in rodents, making them easier prey for predators required to complete the parasite's life cycle. Viruses can disrupt memory, mood, and perception by altering neural chemistry. The brain, rich in nutrients and electrical activity, became valuable territory in the evolutionary struggle. Building on this pattern, evolutionary psychologist Marco del Giudice proposed that parasite pressure may have influenced the growth of increasingly complex brains in primates and humans. The principle is grounded in survival. A nervous system vulnerable to manipulation becomes dangerous to its owner. Organisms better able to detect infection, resist interference, or compensate for neurological damage would possess a major evolutionary advantage. One response may have been the strengthening of barriers protecting neural tissue. The blood-brain barrier acts as a tightly controlled filter between circulating blood and the brain itself. Specialized cells restrict which substances can pass into delicate neural environments. Many pathogens moving through the body never reach the brain because this defensive boundary blocks them before entry becomes possible. Complexity may also have served as protection. Simple systems are easier to disrupt than layered ones. As brains became more interconnected and modular, damage to one region became less likely to collapse the entire structure. Functions could redistribute through overlapping networks. Evolution may therefore have favored redundancy because a more distributed brain is harder for parasites to fully hijack. Neural signaling itself also became increasingly elaborate. Human cognition depends on dense interactions between neurotransmitters, synchronized electrical patterns, and specialized communication pathways spread across many regions at once. The greater this complexity became, the more difficult it may have been for pathogens to imitate or intercept neural communication effectively. Evidence for this broader pattern appears in comparative genetics. Across humans and great apes, immune related genes show some of the strongest signatures of positive selection in the genome. The immune system evolved rapidly under intense pressure, but many immune pathways are closely linked to brain development and nervous system maintenance. Inflammation, neural repair, and immune signaling overlap continuously inside living tissue. The brain and immune system did not evolve separately. They adapted together under many of the same pressures. This possibility reshapes the meaning of human cognition in subtle ways. Memory, emotional regulation, abstract reasoning, and self awareness may not exist solely because they improved hunting or social coordination. Some aspects of mental complexity may also reflect ancient defensive architecture, biological systems shaped partly to resist infiltration and behavioral manipulation. Seen through this lens, the human mind becomes more than a tool for thought. It resembles a carefully guarded environment, refined across millions of years against threats too small to see directly. Beneath language, imagination, and reflection lies a nervous system carrying traces of an invisible conflict, one that may have helped shape the very structure of consciousness itself. When small groups of modern humans began leaving Africa roughly 60,000 to 70,000 years ago, they entered environments already inhabited by another human species. Neanderthals had lived across Eurasia for at least 200,000 years, surviving glacial cold, unfamiliar parasites, and microbial ecosystems completely different from those of Africa. Their bodies had already spent thousands of generations adapting to local diseases long before Homo sapiens arrived. For the newcomers, the danger was not limited to climate or terrain. Every region carried its own invisible biological landscape. Pathogens that Neanderthals encountered routinely may have been entirely unfamiliar to migrating human populations. An immune system shaped in Africa suddenly faced bacteria, parasites, and viruses it had never experienced before. At some point during this long overlap, the two human lineages interbred. The encounters were rare enough that Neanderthals eventually disappeared as a distinct species, yet common enough to leave a permanent mark inside the modern human genome. Today, most people of non African ancestry carry between 1 and 4% Neanderthal DNA inherited through generations, stretching back tens of thousands of years. What surprised researchers was not simply the existence of this inherited DNA, but where it remained concentrated. Many Neanderthal sequences disappeared over time, gradually removed through natural selection or genetic drift. Yet certain regions persisted strongly across Eurasian populations, especially in areas connected to immune function. Among the most important are genes involved in toll-like receptors, usually abbreviated as TLRs. These receptors function as one of the immune system's earliest warning mechanisms. They recognize common molecular signatures associated with invading pathogens and trigger rapid defensive responses before infections spread deeply through the body. Several Neanderthal derived TLR variants appear at unusually high frequencies in modern Eurasian populations. Studies suggest these inherited versions increased immune sensitivity, allowing the body to detect and respond to pathogens more aggressively. For populations entering unfamiliar disease environments, such heightened reactivity likely carried substantial survival advantages. Further evidence emerged through genomic research examining interactions between human proteins and RNA viruses. A study published in Cell found that regions associated with viral interaction were strongly enriched for Neanderthal ancestry in European populations. The pattern suggests that ancient viral pressures shaped which Neanderthal genes survived inside modern humans. Some inherited immune variants may have spread precisely because they improved resistance against Eurasian viruses already circulating long before Homo sapiens arrived. The exchange was, therefore, not merely biological mixing between two human groups. It functioned partly as an evolutionary transfer of local immune knowledge encoded directly into DNA. Modern humans entering Eurasia acquired genetic adaptations that Neanderthals had spent hundreds of thousands of years accumulating under regional pathogen pressure. Yet, the benefits carried consequences that remain visible today. Many Neanderthal immune variants associated with stronger pathogen responses are also linked to elevated risks of allergies and inflammatory disorders in modern populations. The same immune sensitivity that once improved survival against ancient infections can now react excessively to harmless substances such as pollen, dust, or food proteins. The immune system evolved for a world saturated with parasites, chronic infections, and continual microbial exposure. In that environment, rapid alarm responses were often beneficial. In modern settings, especially in industrialized societies with reduced pathogen load, some of those inherited responses no longer match the conditions surrounding them. Even now, traces of those ancient encounters persist quietly within living people. Embedded among the 3 billion base pairs of the human genome are fragments inherited from another species of human, preserved not by accident, but because they once helped unfamiliar bodies survive unfamiliar diseases in a foreign world. In October of 1347, trading ships entered the harbor of Sicily, carrying more than silk, grain, and cargo from the east. Hidden among rats and fleas traveling inside the wooden hulls was a bacterium called Yersinia pestis. Within a few years, the disease it unleashed spread across Europe, North Africa, and the Middle East with extraordinary speed, killing between 30 and 50% of the population. Entire towns emptied. Trade collapsed. Burial grounds expanded faster than cities could manage them. The plague moved through the body brutally. Swollen lymph nodes darkened beneath the skin. Fever climbed rapidly. Blood vessels ruptured. Septic shock overwhelmed organs, while tissue began dying from within. In some forms of infection, the lungs filled with bacteria dense enough to spread directly through coughing. A healthy person could develop symptoms and die within days. For centuries, the Black Death was understood mainly as a demographic catastrophe, a historical rupture visible in abandoned villages, labor shortages, and cultural upheaval. Yet, beneath the visible destruction, another process unfolded quietly inside the surviving population. Natural selection accelerated. In 2022, researchers from McMaster University, the University of Chicago, and the Institut Pasteur analyzed ancient DNA from more than 200 individuals buried in London and Denmark before, during, and after the plague years. By comparing genomes across these periods, they searched for signs that the epidemic had altered the genetic structure of surviving populations. The evidence pointed towards several immune-related genes undergoing unusually strong positive selection during the Black Death. Certain variants became significantly more common among survivors, suggesting they improved resistance against infection strongly enough to influence who lived long enough to reproduce after the epidemic passed. Among the most striking examples was a gene called ERAB2. This gene helps immune cells process and present fragments of invading pathogens, allowing the immune system to recognize danger more effectively. Individuals carrying a particular variant of ERAP2 appear to have survived plague infection at substantially higher rates than those lacking it. Over time, that protective variant spread widely through European populations. The speed of this change matters. Evolution is often imagined as unfolding too slowly for human observation, drifting almost invisibly across immense spans of time. The Black Death demonstrated that, under extreme pressure, the human genome can shift measurably within only a few generations. A single pathogen, operating for less than a century, altered the distribution of immune related genes across millions of people. Yet the adaptation carried consequences beyond the medieval world that produced it. The same ERAP2 variant associated with plague survival is now linked to elevated risk for Crohn's disease and rheumatoid arthritis. A defensive system calibrated for an environment saturated with lethal infection continues operating inside bodies living under very different conditions. The immune system does not evolve toward comfort or balance in any permanent sense. It evolves toward immediate survival under the pressures present at the time. During the plague years, heightened immune reactivity improved the odds of surviving bacterial invasion. In modern environments with reduced exposure to the same pathogens, those inherited responses can become excessive, turning inflammation against healthy tissue. The Black Death therefore became more than a historical disaster. It served as a direct demonstration of evolutionary pressure acting on human populations in real time. Beneath the chronicles of mass graves and collapsing cities, invisible genetic selection moved through the survivors, quietly reshaping the biology carried forward into future generations. Buried within chromosome 6 is one of the most crowded and variable territories in the entire human genome. This region, known as the major histocompatibility complex, stretches across roughly 3.6 million base pairs and contains some of the highest genetic diversity ever observed in human biology. No two people carry exactly the same arrangement unless they are identical twins. The importance of this diversity becomes clearer when looking at what these genes actually do. MHC molecules sit on the surfaces of cells like tiny display platforms, continuously presenting fragments of material taken from inside the cell itself. Most of these fragments are ordinary proteins produced during normal cellular activity, but if a virus, bacterium, or parasite has entered the cell, pieces of the invader may also appear among them. Immune cells constantly inspect these molecular displays. When the presented fragments appear familiar and harmless, the immune system remains inactive. When unfamiliar or dangerous material appears, defensive responses begin rapidly. The MHC therefore functions as part of the body's identification system, allowing immune cells to distinguish healthy tissue from infection through continuous molecular surveillance. What makes this system extraordinary is the scale of variation across the human population. Thousands of different MHC alleles exist globally, distributed unevenly among populations shaped by different pathogen environments over thousands of generations. Certain variants became common where specific diseases exerted strong pressure, while others remained rare or localized to particular regions. This immense diversity persists because uniformity would be dangerous. If every individual carried identical MHC variants, a pathogen capable of bypassing one immune profile could potentially spread through entire populations with devastating efficiency. Diversity itself became a defensive strategy at the species level. Two evolutionary mechanisms help maintain this variation. The first is heterozygote advantage. Individuals inheriting two different MHC alleles can recognize a broader range of pathogens than those carrying two identical versions. Their immune systems effectively possess a wider surveillance library, increasing the probability that invading organisms will be detected before infection becomes overwhelming. The second mechanism emerges through continuous evolutionary feedback between pathogens and hosts. When a particular MHC variant becomes extremely common, pathogens experience pressure to evade recognition by that specific immune profile. Over time, rare MHC variants may suddenly become advantageous because pathogens are less adapted to bypass them. As those rare variants spread, selective pressure shifts again. The balance never stabilizes permanently. Host and pathogen continue adjusting to one another across generations. This pattern reflects one of the clearest examples of Red Queen dynamics inside the genome itself. The advantage belongs not to a permanently superior immune variant, but to whichever configuration remains slightly less predictable to evolving pathogens at a particular moment in time. The evolutionary roots of the MHC extend deep into ancient life. Elements of this molecular recognition system predate modern vertebrates and have remained under intense pathogen driven selection for more than 500 million years. Long before mammals emerged, organisms were already evolving methods for distinguishing self from invader with increasing sophistication. Even now, the MHC continues changing quietly across human populations. Every epidemic, migration, and environmental shift alters the selective landscape surrounding immune recognition. Beneath ordinary biological function, these genes preserve the memory of ancient microbial pressures while continuing to adapt to new ones. The result is a genomic region shaped not toward perfection, but toward constant flexibility, an evolving catalog of defensive possibilities maintained through endless biological negotiation with the microscopic world. Attraction often feels immediate and difficult to explain. A person enters a room, and something about them seems quietly compelling long before conscious thought begins assembling reasons. Facial expression, voice, posture, and movement all contribute. Yet part of the response may emerge from a far older biological system operating beneath awareness, one shaped by the evolutionary struggle against disease. Research into the major histocompatibility complex revealed that human attraction is influenced partly by immune genetics. The discovery emerged most famously through an experiment conducted in 1995 by Swiss biologist Klaus Wedekind. In what later became known as the sweaty t-shirt study, men wore plain cotton shirts for several nights under controlled conditions, avoiding scented products and foods that might alter body odor. Women were then asked to smell the shirts and rate the attractiveness of the scents. The pattern that emerged was striking. Women consistently preferred the odor of men whose MHC profiles differed from their own. Without seeing faces or hearing voices, participants appeared capable of detecting genetic compatibility through smell alone. The mechanism is subtle and indirect. MHC genes influence the chemical environment of the body, shaping how proteins break down and interact with skin bacteria. Those bacterial communities then produce volatile compounds released through sweat and skin oils. The resulting scent carries faint information about immune system genetics, encoded chemically rather than consciously. The nose does not interpret this information in deliberate terms. No one smells another person and consciously identifies immune diversity. Yet, the nervous system appears capable of responding to these chemical cues automatically, guiding attraction toward genetically dissimilar immune profiles. The evolutionary logic is remarkably efficient. Offspring inheriting two different sets of MHC variants possess a broader ability to recognize pathogens. A more diverse immune system increases the range of bacteria, viruses, and parasites the body can detect effectively. Over thousands of generations, attraction patterns favoring immune diversity would therefore improve survival rates among descendants. The influence of the MHC extends beyond scent alone. Research suggests these genes affect olfactory receptor expression, aspects of pregnancy compatibility, and possibly visual perception of attractiveness. A study published in 2005 found correlations between MHC heterozygosity and ratings of facial symmetry, suggesting that immune diversity may subtly shape developmental stability across the body. One of the most intriguing findings involves hormonal contraceptives. Women using oral contraceptives often show weakened or reversed MHC scent preferences compared to naturally cycling participants. Because hormonal contraceptives partially simulate aspects of pregnancy physiology, researchers proposed that the body may temporarily shift toward preferring familiar or genetically similar social cues during altered hormonal states. The implications extend quietly into modern life. Mate choice, long assumed to be driven mainly by culture, personality, or conscious preference, also carries biological influences inherited from ancient pathogen pressure. Beneath romance and familiarity, the nervous system continues, evaluating signals tied to immune compatibility. This does not reduce human attraction to chemistry alone. Human relationships remain shaped by memory, culture, emotion, and individual experience. Yet biology still participates in the process through mechanisms refined across immense spans of evolutionary time. The arms race against pathogens reached deeply enough into human evolution that it influenced not only immune defenses, but the sensory systems involved in choosing partners. Hidden beneath ordinary perception, the body still searches for genetic combinations that might give future offspring a slightly better chance against the microbial world surrounding them. A primate living alone does not need to remember very much about other minds. Survival depends mostly on food, shelter, danger, and movement through the environment. But once social groups become larger and more complex, a different kind of pressure begins accumulating inside the nervous system. Every individual becomes part of a shifting network of alliances, rivalries, favors, trust, and deception. Social life itself turns into a cognitive landscape that must be continuously mapped. During the 1990s, evolutionary anthropologist Robin Dunbar examined this problem by comparing brain structure across primate species. His findings revealed one of the most consistent patterns in comparative neuroscience. Species living in larger and more socially demanding groups tended to possess proportionally larger neocortices, the outer layers of the brain associated with higher cognition, planning, memory, and abstract processing. The relationship was not based on total brain size alone. Large bodies often require larger brains simply to coordinate muscles and sensory systems. What mattered in Dunbar's analysis was the neocortex ratio, the volume of the neocortex relative to the rest of the brain. Across primates, this ratio predicted average social group size with remarkable precision. The implication quietly reshaped theories about human intelligence. Brain expansion may not have occurred primarily because early humans needed to navigate landscapes, craft tools, or locate food. Those abilities mattered, but the strongest pressure may have come from other humans. Managing relationships inside increasingly large groups required enormous cognitive effort. A chimpanzee living among 50 individuals faces a constant stream of social calculations. It must remember who supported whom during conflicts, who shares food, which alliances remain stable, which individuals are dominant, and which rivalries are becoming dangerous. Social memory becomes essential for survival. A single mistake in reading group dynamics can alter access to mates, protection, or resources. This cognitive capacity is often described as theory of mind, the ability to understand that other individuals possess thoughts, intentions, and desires separate from one's own. A socially intelligent primate does not merely observe behavior, it predicts motives, it interprets attention, expectation, loyalty, and deception. Such processing requires vast neural coordination occurring continuously beneath ordinary interaction. As group size increases, the difficulty expands rapidly. Each new relationship multiplies the number of possible alliances and conflicts within the network. Intelligence becomes less about solitary problem solving and more about maintaining stable social positioning inside a crowded mental landscape. Using the relationship between neocortex ratio and group size, Dunbar estimated that humans possess a natural upper limit for stable social relationships at around 148 individuals, now commonly known as Dunbar's number. The figure appears repeatedly across human societies, hunter-gatherer communities, military company structures, and many traditional social organizations often stabilize around similar sizes before fragmentation begins. The pattern suggests that even modern civilization still operates partly within limits established by ancient neural architecture. Human cognition evolved in environments where survival depended on tracking manageable social networks rather than interacting with thousands of strangers simultaneously. The social brain hypothesis therefore reframes the origins of intelligence itself. The expanding human mind may have emerged not simply through interaction with the physical world, but through continuous negotiation with other minds equally capable of strategy, memory, cooperation, and manipulation. Long before cities, writing, or technology appeared, evolution had already begun constructing a brain shaped by the demands of living among increasingly complicated social worlds. Inside a primate group, survival depends on far more than strength or speed. Every interaction carries hidden information. Who is allied with whom? Who can be trusted? Who is watching? Who is pretending not to watch? All of it shapes access to food, protection, status, and reproduction. Social life becomes a continuous exercise in prediction. In 1988, primatologists Andrew Whiten and Richard Byrne proposed that much of primate intelligence evolved specifically to manage this environment of strategic social competition. They called the idea Machiavellian intelligence, borrowing the name from Niccolò Machiavelli, whose political writings focused on manipulation, strategy, and power within human societies. Their argument began with a simple observation. Species possessing larger neocortices also tended to display more frequent and sophisticated forms of tactical deception. Among chimpanzees, for example, subordinate individuals sometimes pretend not to notice food desired by dominant group members. They glance away, appear uninterested, and wait until attention shifts before quietly retrieving it. Such behavior requires more than instinct. The animal must model what another individual can see, what they know, and how they are likely to react. This type of cognition creates a self-reinforcing evolutionary cycle. If one member of a group becomes better at deception, others face pressure to improve deception detection. As detection improves, more subtle forms of manipulation become advantageous. The cognitive baseline of the group gradually rises because social competitors continuously adapt to one another. Unlike environmental challenges, social challenges evolve in response. A river does not become harder to cross because an animal learned to swim. Another mind does. Every successful strategy alters the behavior of surrounding individuals, forcing further adaptation throughout the group. Humans carried this process to extraordinary levels. Daily social life depends on tracking relationships extending far beyond immediate family or direct interaction. Reputation spreads through conversation. Alliances form and dissolve across networks containing dozens or hundreds of individuals. People remember favors, detect insincerity, anticipate reactions, and adjust behavior depending on audience and status. Much of human conversation revolves around social information itself. Gossip functions partly as reputation management, allowing groups to exchange data about trustworthiness, loyalty, cooperation, and threat. Information becomes social currency, shaping how individuals are treated long before direct experience occurs. The cognitive demands underlying this system are immense. Humans routinely construct nested mental models about what others believe. A person may consider not only what someone thinks, but what that person believes another individual thinks in return. This recursive layering is sometimes described as fifth order intentionality. Very few other species demonstrate evidence for comparable levels of abstraction. The result is not merely a larger primate brain, but a qualitatively different style of cognition. Human intelligence became deeply specialized for navigating social complexity, memory, language, emotional interpretation, and attention all intertwined with the demands of group living. This perspective changes the meaning of intelligence itself. Many features of the human mind may not have emerged primarily for solitary reasoning or environmental mastery. They developed because survival increasingly depended on understanding other minds operating nearby, minds capable of cooperation, rivalry, concealment, persuasion, and strategic calculation. The pressure shaping this cognitive escalation did not come mainly from predators, storms, or changing landscapes. It emerged from individuals living side by side within the same social world, each adapting continuously to the behavior of everyone around them. Not every trait in evolution exists because it improves survival directly. Some survive because they attract attention. A peacock's tail is heavy, brightly colored, difficult to hide, and energetically expensive to maintain. From a purely practical perspective, it appears inefficient. Yet generation after generation, peahens selected males carrying larger and more elaborate tails, allowing the trait to expand far beyond simple utility. In 2000, evolutionary psychologist Jeffrey Miller proposed that part of the human brain may have evolved through a similar process. In The Mating Mind, he reframed human intelligence around Darwin's second major evolutionary mechanism, sexual selection. The argument addressed a long-standing puzzle surrounding the human brain. Many aspects of human cognition seem excessive for the basic demands of hunter-gatherer survival. The brain consumes enormous amounts of energy, roughly 20% of the body's calories while at rest, despite representing only a small fraction of total body mass. Maintaining such tissue is biologically expensive, yet humans developed capacities extending far beyond immediate survival needs. Music, humor, storytelling, artistic symbolism, abstract mathematics, and philosophical thought do not obviously increase the ability to escape predators or locate food on a savanna. Miller suggested that these traits may have functioned partly as courtship displays. Intelligence became visible through behavior. Creativity, wit, memory, verbal skill, emotional insight, and problem-solving ability all signaled the quality of the underlying nervous system producing them. The The process likely unfolded gradually across generations. If ancestral females showed preference for males, displaying greater cognitive flexibility through storytelling, social intelligence, humor, or innovation, then those males reproduced more successfully. Their offspring inherited both the traits being selected and the preferences favoring those traits. Over time, the feedback loop intensified. This mechanism is known as Fisherian runaway selection, named after geneticist Ronald Fisher. Once a preference and a trait become genetically linked within a population, both can escalate together. Selection no longer stabilizes around practical necessity alone. Traits may continue growing more elaborate because they remain desirable to potential mates. Under this framework, human intelligence became partly self amplifying. Individuals competed increasingly through displays of cognitive ability rather than physical ornament alone. Language, music, narrative skill, symbolic thought, and social performance all became arenas of reproductive competition inside the species itself. The timescale of human brain expansion adds weight to the argument. Brain size roughly tripled over approximately 2 million years, a remarkably rapid transformation in evolutionary terms. Few other explanations account simultaneously for both the speed and apparent excess of human cognition. Survival pressures alone struggle to explain why the brain developed capacities extending so far beyond immediate ecological demands. Sexual selection offers a possible mechanism for that escalation. Once minds themselves became objects of selection, evolution began favoring increasingly elaborate displays of mental ability. Cognitive sophistication turned into a reproductive advantage. The competition unfolding inside human groups therefore changed form. Earlier evolutionary pressures favored physical survival against predators, pathogens, and environmental instability. Sexual selection added another layer, one centered on attraction, performance, and social impression. Each generation inherited nervous systems shaped partly by the preferences of the generation before it. The result was a species whose most elaborate biological structure may have emerged not solely because it solved problems, but because it impressed other minds. Beneath poetry, music, humor, and imagination lies an ancient evolutionary tournament sustained across countless generations through the subtle and continuous pressures of attraction itself. A spoken sentence can alter trust, reshape alliances, end conflicts, start wars, preserve memories, or destroy reputations without any physical contact at all. Among all human adaptations, language became one of the most powerful tools ever produced by evolution, not only because it allowed cooperation, but because it transformed social competition into something far more complex than physical dominance alone. Robin Dunbar proposed that language may have originated partly as a replacement for social grooming. Among primates, grooming strengthens alliances, reduces tension, and maintains group cohesion. Yet grooming has strict limits. One individual can groom only one partner at a time, and maintaining large social networks through touch alone becomes increasingly inefficient as group size expands. Speech changed that limitation entirely. A single speaker could interact with several individuals simultaneously, reinforcing social bonds across much larger communities. Words allowed information, emotion, and social reassurance to move rapidly through groups without direct physical contact. Language became a scalable form of social connection. Much of human conversation still reflects this ancient function. Across cultures, a large percentage of ordinary speech concerns third parties rather than immediate survival tasks. Researchers estimate that roughly 65% of conversation involves gossip, meaning the exchange of social information about absent individuals. This is not meaningless chatter. Gossip monitors trust, reputation, loyalty, and status inside groups. Through language, individuals can warn others about deception, reinforce cooperation, spread admiration, damage rivals, or strengthen alliances. Reputation becomes a shared social construct maintained collectively through repeated communication. A person's standing inside the group increasingly depends not only on direct interaction, but on stories circulating beyond their immediate presence. Once language emerged, social competition changed dramatically. Individuals skilled at persuasion, storytelling, emotional framing, or strategic communication gained advantages extending far beyond physical strength. Influence could now spread indirectly through narrative itself. This created new evolutionary pressures operating inside human societies. Producing convincing speech became valuable, but so did interpreting it accurately. Listeners capable of detecting dishonesty, sarcasm, manipulation, or hidden intention avoided social costs more effectively than those who accepted communication at face value. Human cognition, therefore, expanded not only toward language production, but toward language analysis. The nervous system adapted to track ambiguity, tone, implication, contradiction, and emotional nuance simultaneously. Understanding speech required modeling the intentions behind words as much as the words themselves. Communication evolved into a layered exchange where meaning often depended on context, memory, reputation, and inferred motive. The biological foundations supporting this transformation appear deep within the human lineage. FOXP2, introduced earlier as one of the genes associated with speech and neural coordination, contains two amino acid changes unique to humans and our closest extinct relatives. Neanderthals carried these same variants, indicating that the capacity for advanced vocal communication likely predates the split between Neanderthals and modern humans by at least 600,000 years. Language, therefore, emerged gradually within an already intensifying social environment. As groups became larger and relationships more complex, communication itself became part of the evolutionary arms race. Every improvement in persuasion increased pressure for better skepticism. Every improvement in deception increased pressure for sharper interpretation. What developed was not simply a species capable of exchanging information, but one capable of constructing entire social realities through words alone. Human language became more than communication. It became a system for shaping alliances, preserving memory, regulating reputation, and navigating the invisible social structures surrounding every human group. Somewhere inside every epidemic lies the same imbalance. A virus can generate thousands of mutations while a single human generation is still being born. Bacteria divide continuously, adapting hour by hour to chemical threats that did not exist the day before. In purely genetic terms, the arithmetic seems hopeless. Human beings reproduce slowly, mature slowly, and carry bodies built on biological systems that cannot rewrite themselves fast enough to match microbial speed. Yet humans survived this imbalance and eventually expanded into nearly every environment on Earth. Evolution's answer was not to accelerate the genome itself. Instead, adaptation began moving through another channel entirely, one capable of changing far faster than DNA. Human survival became increasingly dependent on culture, memory, and learned behavior transmitted between individuals rather than encoded directly into genes. Fire altered the microbial landscape surrounding early humans long before anyone understood pathogens. Cooking reduced parasite load inside food. Smoke changed insect behavior around campsites. Heat sterilized water and meat accidentally, transforming daily survival through repeated practice rather than biological mutation. Shelter introduced another layer of defense. Walls separated bodies from weather, scavengers, contaminated water, and disease carrying insects. Clothing protected damaged skin from infection. Burial rituals, avoidance practices surrounding corpses, and taboos concerning bodily fluids appeared independently across many cultures, often long before written medicine emerged. These behaviors likely functioned as what evolutionary researchers sometimes describe as a behavioral immune system, a network of inherited social responses, reducing exposure to infection without requiring conscious scientific understanding. Unlike genes, culture could spread almost immediately. A dangerous water source needed to kill only a few individuals before knowledge of it could circulate through the group. A poisonous plant required only a small number of fatal mistakes before stories, rituals, or prohibitions preserved the lesson for future generations. The adaptive immune system, described earlier, already represented one partial solution to the speed asymmetry by generating variation inside a single lifetime. Antibodies diversified rapidly enough to confront pathogens the genome had never encountered before. Culture extended this principle outward, transforming entire human communities into collective learning systems. A group that remembered which wounds became infected, which seasons carried illness, or which animals transmitted disease, possessed a survival advantage unavailable to isolated individuals. Behavior could adjust within years rather than evolutionary millennia. Human populations therefore began adapting socially at speeds biology alone could never achieve. This may also help explain one of the strangest features of human development, the extraordinary length of childhood. Compared to most mammals, human juveniles remain dependent for an unusually long period. Reproduction is delayed while the brain absorbs immense quantities of information from older members of the group. During this extended developmental window, children inherit far more than language or social identity. They absorb survival systems accumulated across generations, including food preparation, hygiene behavior, medicinal knowledge, social rules surrounding contamination, and patterns of collective caution shaped by ancient disease pressures. The result is a species whose primary adaptive weapon is no longer genetic speed alone. Humans evolved the capacity to store solutions outside the body itself, preserving them in memory, ritual, teaching, and shared behavior. Culture became an extension of the immune system, operating across entire populations, allowing a slow reproducing primate to survive inside a biological arms race that should, by every ordinary measure, have overwhelmed it long ago. Long before scientists understood what a virus was, the human genome had already been recording encounters with them for millions of years. Hidden inside chromosomes are traces of ancient conflicts, preserved not as stories or fossils, but as statistical distortions in the rate genes changed over time. Some regions remained almost untouched across evolutionary history. Others shifted repeatedly, as though struck again and again by the same invisible pressure. In 2016, evolutionary biologists studying comparative genomics identified one of the clearest molecular signatures of this pressure ever observed. They examined a category of human proteins known as virus interacting proteins, or VIPs, molecules physically contacted, exploited, or manipulated by viruses during infection. These proteins sit at the exact boundary where viral replication collides with cellular defense. The result was striking. Genes associated with VIPs showed evidence of positive selection at rates approximately 3.9 times higher than genes unrelated to viral interaction. Across evolutionary time, the portions of the genome most exposed to viruses had been reshaped continuously and disproportionately by natural selection. The pattern carried a particular rhythm familiar from every earlier stage of the arms race. A virus evolved a method for entering or hijacking a cell. Human proteins changed in response. Viral evolution adapted again. The cycle repeated over thousands of generations, leaving measurable acceleration inside the genome itself. What emerged was not the signature of occasional epidemics, but of constant biological pressure operating over immense spans of time. Viral conflict was not an interruption to human evolution. It was one of its permanent environmental conditions. This evidence connected directly with another discovery already woven into the human story. Many virus interacting proteins appear enriched within Neanderthal derived sequences preserved in modern Eurasian populations. The fragments of Neanderthal DNA retained after interbreeding were not random remnants scattered passively through the genome. They clustered around regions deeply involved in viral defense. Ancient encounters between Homo sapiens, Neanderthals, and Eurasian pathogens therefore left overlapping genetic footprints. Some inherited Neanderthal variants likely improved survival against unfamiliar viruses encountered outside Africa, allowing those sequences to persist while others vanished. The pattern extended even further into recent human history. In 2018, researchers studying East Asian populations identified signals of positive selection near coronavirus interacting proteins within the last 25,000 years. The findings suggested that an ancient coronavirus or closely related viral epidemic had exerted enough pressure to leave detectable marks in the genomes of its survivors. The modern pandemic revealed how unfinished this process remains. During the spread of COVID-19, researchers identified genetic regions associated with both heightened vulnerability and partial protection against severe disease. Several of these mapped near genes such as CCR5 and OAS1, both connected to Neanderthal introgression and long-standing antiviral defense pathways. The implications moved with quiet force through evolutionary biology. Ancient epidemics were not isolated tragedies fading into the past. Their pressure remained embedded inside living populations, influencing immune responses tens of thousands of years later. Beneath every human cell lies a genomic archive shaped by organisms too small to see directly. Viruses altered the architecture of proteins, redirected selection across entire populations, and helped determine which genetic lineages continued forward through time. The arms race never paused long enough for stability to emerge. It simply descended deeper from visible disease into the molecular machinery of life itself. There are moments in evolutionary history when survival narrows suddenly, as though the future of a species passes through the eye of a needle. Around 74,000 years ago, one of those moments may have arrived in fire. On the island now called Sumatra, the Toba supervolcano erupted with a force difficult to place beside ordinary geological events. An estimated 2,800 cubic kilometers of material blasted into the atmosphere. Ash drifted across continents. Sulfur aerosols spread through the upper sky, reducing sunlight and altering global climate patterns. Temperatures likely fell for years. Forests contracted, food chains destabilized. Rainfall patterns shifted across enormous distances. The eruption remains the largest known volcanic event of the past 2 million years. Some researchers believe humanity passed through a severe population bottleneck during or near this period. Genetic evidence points toward an ancient reduction in human numbers so extreme that the breeding population may have fallen somewhere between 10,000 and 30,000 individuals worldwide. The exact relationship between TOBA and this contraction remains debated, yet the genomic signal itself persists with unusual clarity. Modern humans carry remarkably low genetic diversity compared with other great apes. Chimpanzees, despite their far smaller modern population size, possess substantially greater variation in many immune related regions of the genome. Somewhere in the past, the human lineage lost enormous amounts of biological diversity. A bottleneck of this scale changes evolution profoundly. When populations collapse, chance begins shaping survival almost as strongly as adaptation itself. Rare immune variants disappear permanently. Entire branches of genetic possibility vanish with the individuals carrying them. What survives afterward is not a complete representation of the species that existed before, but a narrow surviving fraction. The consequences extend far beyond simple numbers. Every human alive today descends from whatever remained after that contraction passed. The immune systems, behavioral tendencies, cognitive traits, and social structures carried by those survivors became disproportionately amplified in all future generations. In this sense, the arms race nearly reset itself. The pressure was not merely environmental. Scarcity intensifies disease vulnerability, weakens populations, disrupts migration patterns, and compresses human groups into smaller, more fragile networks where survival depends increasingly on cooperation, memory, and adaptability. Under those conditions, even small biological advantages can determine which lineages continue. The reduced diversity of the human MHC system appears consistent with such an event. Compared with other primates, humans possess unusually limited variation in portions of the genome responsible for pathogen recognition. Some of the immunological flexibility once present in earlier human populations may simply have disappeared during this narrowing, yet survival carried its own strange momentum. A smaller surviving population also meant that advantageous traits could spread more rapidly through subsequent generations. Social coordination, flexible behavior, cultural learning, and cooperative adaptation may have become even more important once genetic diversity contracted. The descendants of that bottleneck eventually expanded outward again, repopulating landscapes emptied by climate instability and time. Every language, civilization, and culture emerged from that thin, surviving thread. Seen from deep time, humanity does not resemble an unstoppable ascent. It resembles repeated passage through conditions that should perhaps have erased it entirely. The genome still carries traces of those constrictions, quiet evidence that the modern human population emerged not from abundance, but from survival after extraordinary reduction. Somewhere in the darkness following Toba, the race continued with only a small remnant left running it forward. Human migration did not unfold across an empty world. Every coastline, forest, river valley, and grassland carried its own invisible ecology of infection long before modern humans arrived there. When small groups began leaving Africa in repeated waves between roughly 60,000 and 70,000 years ago, they entered landscapes already occupied not only by animals and rival human species, but by entire communities of parasites, bacteria, and viruses shaped by deep local history. The movement itself likely followed shifting climates and narrow ecological corridors through the Middle East, then outward into Eurasia, South Asia, Oceania, and eventually the Americas. Yet every step into unfamiliar territory exposed migrating populations to pathogens their immune systems had never encountered before. A parasite adapted to tropical wetlands behaved differently from one thriving in cold northern forests. Mosquito borne diseases concentrated near stagnant water. Intestinal parasites varied with local animals and climate. Viruses circulating among dense populations of Eurasian mammals presented challenges absent from earlier African environments. Each migration, therefore, triggered a new phase of the arms race. The response unfolded gradually inside the genome. Human populations settling in different regions accumulated distinct immune adaptations calibrated to their local pathogen landscapes. Over thousands of years, natural selection reshaped immune related genes differently across continents, producing patterns still visible today. This is one reason immune gene variation between populations is among the highest of any major category in the human genome. The selective pressures acting on skin color, height, or metabolism were significant, but pathogen pressure remained relentless and immediate. A population unable to resist local disease could disappear within generations. The diversity of HLA alleles, the human form of the MHC system described earlier, preserves much of this history. Certain variants associated with resistance to malaria became especially common in sub-Saharan Africa, where mosquito-transmitted parasites exerted continuous evolutionary pressure. In East Asia, other HLA distributions correlate with local, viral, and bacterial histories unique to that region. Similar regional patterns appear throughout indigenous populations of the Americas, Oceania, and northern Eurasia. The genome, therefore, became a geographic record of survival. Every migration route carried biological consequences that accumulated over time, shaping the immunological architecture of entire populations. Yet migration never fully separated these evolutionary pathways. Human groups continued meeting, exchanging genes, competing, and interbreeding across shifting borders. The arms race remained local in pressure, but global in movement. In the modern era, these once separated immunological histories are mixing at unprecedented speed. Air travel, urbanization, global trade, and mass migration now connect populations that spent tens of thousands of years adapting to partially isolated pathogen environments. Diseases cross continents within days. Genetic lineages long separated now combine continuously inside modern cities. Researchers are only beginning to understand the immunological implications of this convergence. Some inherited immune variants may interact in unexpected ways. Certain combinations could broaden pathogen resistance, while others may influence autoimmune risk, inflammatory disorders, or responses to emerging infections. The ancient migration out of Africa, therefore, never truly ended. Its consequences continue unfolding inside living populations, carried invisibly through immune genes shaped by climates, epidemics, and survival struggles stretching across continents and deep time. Every modern human body still carries traces of the particular path its ancestors followed through that long expansion into an already occupied biological world. A human skull carries more than bone. It carries traces of negotiation, conflict, cooperation, and the changing rules of survival written slowly across hundreds of thousands of years. Paleoanthropologists studying skulls from different periods of human evolution noticed that something subtle but persistent had been happening across deep time. The face itself was changing direction. Earlier members of the human lineage often possessed heavier brow ridges, thicker facial bones, and more projecting features. These structures appear strongly associated with testosterone linked development and physical dominance signaling, traits still visible to varying degrees across many primates today. Yet, over roughly the last 300,000 years, the fossil record reveals a gradual shift toward rounder skulls, flatter faces, reduced brow ridges, and greater facial variability. The change did not happen suddenly. It unfolded generation by generation, quietly altering the appearance of the species. In 2017, researchers publishing in Nature, Ecology, and Evolution proposed that this transformation may reflect declining testosterone levels across the human lineage. Their argument suggested that the pressures shaping social success had changed. As human groups became larger, denser, and cognitively more complex, raw physical intimidation may have become less advantageous than social coordination, emotional regulation, and cooperative signaling. In smaller groups dominated primarily by direct competition, physical aggression can determine access to resources, mates, and survival. But larger social systems introduce different pressures. Alliances matter. Reputation matters. The ability to maintain trust across many individuals becomes increasingly valuable. Violence carries social costs, capable of destabilizing the very networks survival depends upon. This fits closely with the broader framework of Machiavellian intelligence explored earlier. The social arms race did not disappear as humans became more cooperative. Instead, it shifted terrain. Success depended less on overpowering rivals physically and more on reading intentions, managing impressions, building coalitions, and navigating fragile social relationships that could change rapidly over time. The human face became central to this transformation. Compared with other primates, humans possess extraordinary facial expressiveness. Roughly 43 facial muscles allow the production of thousands of subtle movements and combinations. Tiny changes around the eyes, mouth, cheeks, and forehead communicate hesitation, suspicion, reassurance, attraction, embarrassment, uncertainty, and countless other social signals often processed almost instantly by the brain. No other primate appears to operate with the same degree of facial nuance. This expressiveness likely emerged because human social life required increasingly precise communication. In a species dependent on cooperation between unrelated individuals, the ability to detect dishonesty, fear, anger, empathy, or intention could directly influence survival. A poorly judged alliance might carry lethal consequences. Misreading aggression or deception inside a tightly interconnected group could destabilize access to protection, food, or reproductive opportunity. The face, therefore, evolved not merely to display emotion, but to transmit information efficiently through complex social environments. At the same time, human perception evolved alongside it. The brain became exceptionally sensitive to facial detail, capable of extracting enormous amounts of social information from slight muscular changes lasting fractions of a second. Entire neural systems specialized in recognizing faces, interpreting gaze direction, and decoding emotional states. Seen across deep time, the modern human face begins to resemble a living social instrument shaped by evolutionary pressure. Bone structure softened while expressive capacity expanded. The battlefield moved away from visible physical dominance and into the subtler terrain of communication, reputation, and emotional interpretation. Long before written language or civilization emerged, survival increasingly depended on the ability to read another human being correctly. For most of human history, the immune system existed inside a world saturated with biological threat. Parasites lived in water, soil, food, insects, and flesh itself. Intestinal worms were so common across ancient populations that many researchers believe the absence of parasites, rather than their presence, is the unusual condition in human history. The immune system that evolved under these conditions was not designed for sterility. It was designed for permanent war. That ancient calibration still exists inside the modern body. In 1989, epidemiologist David Strachan proposed what became known as the hygiene hypothesis. Examining patterns of childhood illness and allergy prevalence, he suggested that reduced exposure to microbes and parasites during development might alter how the immune system matures. The argument was not that cleanliness itself was harmful, but that the immune system evolved, expecting constant biological stimulation, especially early in life. Without those signals, regulation begins to drift. Across industrialized societies, autoimmune disorders and allergies increased dramatically during the second half of the 20th century. The speed of the increase is crucial. Human genes do not transform across a few generations quickly enough to explain such a rapid shift. Something environmental changed first. Urbanization intensified. Antibiotics became widespread. Water systems improved. Chronic parasitic infections declined sharply across many regions. Childhood exposure to diverse microbes narrowed. The immune system remained largely the same system built for another world. Evidence of this mismatch appears repeatedly across the genome. Earlier, the story of ARAP2 revealed how a gene variant associated with survival during the Black Death persists in modern populations today. During plague conditions, heightened immune responsiveness increased the probability of surviving infection. In the modern environment, that same heightened responsiveness is associated with elevated risk for Crohn's disease and rheumatoid arthritis. An adaptation forged under medieval epidemic pressure became, centuries later, a source of chronic inflammation. The same pattern appears in Neanderthal derived toll-like receptor variants retained in Eurasian populations. These variants strengthened early pathogen detection and immune activation, advantages in environments dense with unfamiliar microbes and recurring disease. Yet several are now associated with increased allergy susceptibility. The immune alarm system reacts intensely even when the threat is harmless, sometimes responding to pollen, dust, or food proteins with disproportionate force. The system behaves as though it still occupies a pathogen-heavy Pleistocene landscape. This paradox reveals something unsettling about evolution itself. Natural selection does not produce perfect systems. It produces systems tuned for specific environments, specific pressures, and specific historical conditions. When those conditions change rapidly, older adaptations may persist beyond their original usefulness. The immune system carries millions of years of accumulated defensive logic. Cells patrol constantly through blood, tissue, and lymphatic channels, searching for danger signatures. Inflammatory pathways activate rapidly because, across evolutionary time, delayed response often meant death. The cost of overreaction was frequently lower than the cost of hesitation. Modern industrial life altered that equation faster than biology could follow. For many people alive today, the ancient enemies have been reduced, filtered, medicated, or sanitized. Yet the immune machinery, shaped by parasites, plague bacteria, and chronic infection, continues operating with the same intensity. It still searches relentlessly for invasion. Sometimes, unable to find the threats it evolved expecting, it redirects that aggression inward. The arms race never truly stopped. In some bodies, it simply lost sight of the battlefield. Long before humans understood bacteria, viruses, or infection, culture had already begun evolving in response to them. The process unfolded invisibly across generations, not through mutations in DNA, but through remembered behavior. A successful practice survived because the people who carried it survived. An unsuccessful one disappeared with the communities that depended on it. The Red Queen dynamic extended beyond biology. Unlike genes, culture can move horizontally. A useful adaptation discovered by one group can spread rapidly to others through imitation, trade, migration, or conquest. Ideas replicate faster than reproduction itself. In this sense, human culture became an evolutionary system layered over the slower machinery of genetics, capable of adapting within years rather than millennia. Some of the earliest evidence of this transformation may lie beside ancient fire. Around 1.8 million years ago, changes begin appearing in the anatomy of Homo erectus. Teeth shrink, jaw muscles reduce, the digestive tract becomes smaller relative to body size. Many anthropologists interpret these shifts as evidence of regular cooking. Fire softened food, increased caloric extraction, and reduced chewing time, but it also transformed the pathogen landscape surrounding early humans. Heat destroys parasites, bacteria, and toxins embedded in meat and plant tissue. A cooked carcass is biologically different from a raw one. Groups that mastered fire altered the arms race without waiting for genetic change. The same pattern appears later in burial behavior. By more than 100,000 years ago, archaeological sites begin preserving increasing evidence of intentional treatment of the dead. Bodies were placed carefully, sometimes covered, sometimes isolated from living spaces. The motivations were likely complex, involving memory, ritual, kinship, and symbolism, yet the biological consequence was practical as well. Corpses carry pathogens. Decomposition contaminates water and soil. Separating the dead from the living reduced exposure to disease within densely social groups. Culture repeatedly solved problems faster than evolution alone could manage. Over time, these behavioral systems became increasingly elaborate. Food preparation rules, cleansing rituals, quarantine practices, and taboos surrounding bodily fluids emerged independently across many societies. People rarely understood the microbiology behind these customs, but natural selection did not require understanding. Practices that reduced disease transmission preserved communities more effectively than practices that did not. Then, in the 19th century, the cultural arms race entered a new phase entirely. The development of germ theory transformed disease from an invisible curse into a biological mechanism. Researchers such as Louis Pasteur and Robert Koch demonstrated that microscopic organisms caused infection. For the first time in human history, the slow reproducing species gained the ability to study the fast reproducing one directly. Vaccination expanded. Surgical sterilization improved survival. Antibiotics altered medicine so dramatically that bacterial infections, which once killed millions, became temporarily manageable. For a brief moment, it appeared the ancient asymmetry might finally be breaking. But pathogens continued evolving. Bacteria reproduce rapidly, exchange genetic material, and adapt under intense selection pressure. Every widespread antibiotic creates a new environmental filter, favoring resistant strains. Methicillin-resistant Staphylococcus aureus emerged from this pressure. Drug-resistant tuberculosis followed. Resistant malaria strains appeared across multiple regions. Hospitals themselves became evolutionary arenas where microbial populations adapted continuously to chemical assault. The pattern remained unchanged beneath the technology. Each human innovation reshaped the battlefield. Each microbial counter-adaptation reshaped it again. The arms race never ended when culture emerged. Culture simply became another weapon carried into it. Hidden inside the human genome are fragments of ancient invasions. They do not resemble bones, footprints, or preserved shells. They are quieter than fossils, buried not in stone, but in DNA itself. Every human cell carries them. Every heartbeat unfolds in their presence. Approximately 8% of the human genome consists of sequences derived from ancient retroviruses, known as endogenous retroviruses, or ERVs. These sequences originated when viruses infected the reproductive cells of distant ancestors and inserted their genetic material directly into the host genome. If the infected individual reproduced successfully, the viral sequence passed into the next generation, then the next, slowly spreading across entire populations over immense stretches of evolutionary time. Most of these sequences are now inactive. Mutations accumulated gradually, damaging the viral instructions until replication became impossible. What remains are genomic remnants, molecular ruins preserved across millions of years. Yet the silence of these sequences is deceptive. Some were not merely tolerated by evolution, they were retained. In certain cases, they were transformed into something useful. One of the most extraordinary examples involves proteins called syncytins, essential for the formation of the placenta in placental mammals. The placenta is an organ of controlled biological fusion. Cells from mother and embryo must connect intimately while remaining genetically distinct. Syncytins make this possible by enabling cellular membranes to merge together, forming the layered tissue that allows nutrients and oxygen to pass between bodies. The remarkable detail is their origin. Syncytins are derived from ancient retroviral envelope proteins, the very machinery viruses originally evolved to fuse with host cells during infection. A weapon once used to invade ancestral tissue became necessary for mammalian reproduction itself. Human pregnancy depends, in part, on molecular tools inherited from ancient viruses. The arms race did not merely leave scars in the genome, it altered the architecture of human biology. Other endogenous retroviral sequences appear to influence immune regulation. Some interfere with viral replication pathways, reducing the ability of related viruses to spread efficiently through cells. In effect, fragments of older viral invasions may help defend against newer ones. The genome preserves captured genetic machinery and redeploys it against future threats. Evolution repeatedly works this way. Structures built for one purpose are repurposed for another. Across deep time, the distinction between enemy and adaptation begins to blur. The human genome is therefore not entirely human in origin. It is a layered historical record assembled through contact, conflict, mutation, and survival. Ancient retroviruses inserted themselves into ancestral DNA, seeking only replication. Instead, some became permanent residents. Others became tools. A few became indispensable. This changes the way the body can be understood. The genome is not a pristine instruction manual descending unchanged across generations. It is a battlefield archive. Embedded within chromosomes are records of infections older than humanity itself, preserved from periods when mammals were small, nocturnal animals moving beneath dinosaur-dominated ecosystems. Some ERV lineages are tens of millions of years old, surviving from evolutionary worlds almost unimaginably distant from the present. Every human being carries this archive silently. Inside each cell exists a molecular history of ancient viral encounters compressed into sequences of nucleotides passed from parent to child across unbroken generations. The arms race with viruses became so prolonged, so intimate, that fragments of the enemy entered the genome permanently and remained there long after the original wars disappeared from the earth. The ancient conflicts that shaped human evolution did not end when civilization emerged. They adapted. The battlefield expanded from forests and rivers into hospitals, megacities, laboratories, livestock farms, and digital networks carrying information across the planet in seconds. Beneath modern life, the same pressures continue moving silently through biology and behavior. The arms race is still active. In 1945, while accepting the Nobel Prize for the discovery of penicillin, Alexander Fleming warned that bacteria would evolve resistance if antibiotics were overused or improperly administered. His concern rested on evolutionary inevitability. Any bacterial population exposed repeatedly to antibiotics would eventually contain rare individuals capable of surviving the treatment. Those survivors would reproduce rapidly, passing resistance into future generations. The warning proved accurate. By 2019, antibiotic resistant infections were estimated to cause approximately 1.27 million deaths annually worldwide. Current projections suggest that by 2050, the number could rise toward 10 million per year if resistance trends continue. Methicillin-resistant Staphylococcus aureus, multidrug resistant tuberculosis, and resistant malaria strains have become major evolutionary fronts where medicine and microbial adaptation continuously collide. Every antibiotic changes the environment surrounding bacteria. Every change creates new selective pressure. At the same time, humans increasingly encounter pathogens originating beyond the species boundary itself. Most emerging infectious diseases begin in animals before crossing into humans through zoonotic spillover. Viruses circulate constantly within bats, birds, rodents, pigs, and countless other reservoirs. As forests shrink, wildlife trade expands, and industrial farming intensifies, contact between species becomes more frequent and more compressed. The biological separation that once slowed transmission weakens. When a pathogen first enters humans from another species, the population possesses little evolved resistance. The early phases of spillover, therefore, become periods of intense evolutionary instability. Some pathogens disappear quickly, others adapt successfully to human transmission and spread through globally connected populations. The COVID-19 pandemic demonstrated how rapidly such transitions can transform everyday life across entire continents simultaneously. Yet the social arms race also persists. Now operating inside technological environments, no human nervous system evolved anticipating. The cognitive architecture shaped for small groups still governs attention, status perception, alliance building, and deception detection. Human brains evolved to navigate social worlds measured in dozens, or perhaps hundreds of individuals. Today, those same neural systems operate inside digital spaces containing millions. Reputation spreads instantly. Conflict scales globally. Social approval and exclusion arrive continuously through algorithms designed to capture attention. Ancient instincts now move through electronic networks. Information itself became part of the competitive landscape. Narratives shape alliances. Public identity functions as social currency. Reputation can expand or collapse within hours. The same cognitive machinery once used to interpret expressions around a campfire now processes endless streams of symbolic interaction. Even sexual selection may still influence the structure of culture. Geoffrey Miller's proposal remains unresolved, yet difficult to dismiss entirely, that many forms of human creativity continue functioning partly as display. Music, humor, literature, philosophy, visual art, and intellectual performance may still carry echoes of the same competitive signaling pressures that shaped the expanding human brain across deep time. The modern world appears technologically unprecedented, yet beneath its surfaces, the evolutionary logic remains ancient. Humanity transformed the environment with extraordinary speed, but the biological systems carrying human thought, fear, attraction, competition, and survival were shaped in a much older world, and they continue responding to pressures that never fully disappeared. Inside every human body exists a history of survival older than language itself. Long before cities, agriculture, or even symbolic thought, countless generations moved through environments filled with predators, parasites, famine, disease, and competition. Most lives vanished without record. Some continued. Every person alive today descends only from those who survived long enough to reproduce, forming an unbroken biological chain extending backward across immense spans of time. Yet, survival in evolution was never absolute victory. No organism becomes permanently safe. Adaptation only grants a temporary advantage against the pressures existing at that particular moment. A useful trait in one environment may later become harmful. A powerful immune response that once protected against infection may later increase the risk of autoimmune disease. Van Valen's Red Queen law describes this clearly. Species must keep adapting, simply to maintain their position against competitors, pathogens, and changing environments that continue evolving alongside them. Each generation entered the race again from the beginning. The consequences of those struggles still exist physically within modern genomes. During the Black Death, enormous portions of Europe, North Africa, and the Middle East died within only a few years. Individuals carrying certain immune variants survived at slightly higher rates, allowing those variants to spread through later populations. Centuries afterward, descendants still carry pieces of that selective event inside their immune systems. Ancient epidemics remain biologically present in living people who have never encountered those diseases themselves. The same pattern stretches further back into deep time. Human populations leaving Africa encountered unfamiliar climates, parasites, and pathogens across Eurasia. Interbreeding with Neanderthals introduced immune adaptations shaped over hundreds of thousands of years in different environments. Viral epidemics altered which genetic variants persisted. Population bottlenecks reduced diversity so severely that entire branches of human variation disappeared forever. Roughly 74,000 years ago, after catastrophic environmental instability, only a narrow, surviving remnant contributed disproportionately to all future humans. 3 billion base pairs carried the survivors forward. The genome is therefore less a perfect blueprint than a layered archive assembled through repeated evolutionary emergencies. Some genes remain because they defended against ancient bacteria. Others persist because they improved social cooperation, reproductive success, or resistance to parasites that no longer dominate the modern world. Even endogenous retroviruses, fragments left by infections millions of years old, remain embedded inside human DNA like molecular fossils from forgotten biological conflicts. The mind itself emerged from these pressures. Fear, attachment, suspicion, attraction, jealousy, empathy, and social awareness developed within nervous systems shaped under continuous competition and cooperation. Human cognition was not constructed outside the arms race, it was refined by it. The ability to interpret expressions, remember alliances, detect deception, and communicate through language all carried survival value within increasingly complex groups. Thoughts that now feel deeply personal were shaped gradually by pressures acting across generations, far older than civilization. None of this history disappeared. It became biology. The deepest paradox of evolution is that every living human represents both survival and extinction simultaneously. Your existence required an uninterrupted chain of successful ancestors, yet it also required the disappearance of countless others whose genes, adaptations, and lineages ended before reaching the present. The arms race did not happen around humanity. It constructed humanity piece by piece, leaving its signatures inside every cell now alive. Seen from deep time, the human species appears less like a singular invention and more like an accumulation of solutions layered one upon another across millions of years. Each adaptation emerged under pressure, tested repeatedly against environments, pathogens, rivals, and shifting social worlds. What exists now is the temporary outcome of countless biological negotiations, stretching back to a small ape lineage that once moved through African forests without any obvious sign of what it would eventually become. The immune system alone carries the complexity of an entire ecosystem. Within the body, billions of immune cells circulate continuously through blood, tissue, bone marrow, and lymphatic vessels, scanning for molecular patterns associated with invasion or cellular failure. Through mechanisms refined over immense spans of evolutionary time, the adaptive immune system can generate billions of unique receptor combinations, each capable of recognizing different pathogenic structures. Memory cells preserve fragments of previous encounters for decades, sometimes for life, allowing the body to respond more rapidly when an old threat returns. Every infection leaves an imprint behind, transforming the immune system into a living archive assembled through experience. The nervous system followed a similarly extraordinary trajectory. The human brain, weighing only a small fraction of total body mass, consumes roughly one-fifth of the body's resting energy supply. Inside that metabolically expensive organ reside approximately 86 billion neurons linked through networks of extraordinary density. Electrical and chemical signals move constantly through an estimated 100 trillion synaptic connections, sustaining language, memory, imagination, planning, abstraction, and self awareness. The ability to construct mathematics, compose music, imagine futures, or reflect internally emerged gradually from nervous systems originally shaped for survival within competitive social groups and unstable environments. The face became part of this evolutionary transformation as well. Human facial musculature expanded into an unusually expressive signaling system capable of producing thousands of distinct combinations of movement. Tiny shifts around the eyes, mouth, forehead, and jaw transmit information about intention, emotion, uncertainty, aggression, trustworthiness, and attention. Social life increasingly depended upon accurately reading these signals. Over time, the face evolved into a high-fidelity communication surface, broadcasting internal states rapidly across complex human groups. Even attraction absorbed the imprint of the arms race. The olfactory system quietly monitors chemical traces linked to immune compatibility, detecting biological information beneath conscious awareness. Preferences that feel personal or emotional often contain ancient evolutionary logic beneath them, shaped partly by pressures favoring offspring with broader pathogen resistance. The history of infection entered courtship itself, embedding survival calculations inside scent, attraction, and reproduction. Yet the most unusual development in the human lineage was the emergence of culture as an adaptive force operating faster than genes. Fire altered digestion and reduced pathogens in food. Language allowed knowledge to survive individual lifetimes. Medicine transformed survival probabilities that biology alone could never achieve. Social memory accumulated across generations, permitting human groups to adapt to threats within decades rather than millennia. Evolution partially escaped the limits of slow genetic inheritance by externalizing adaptation into tools, symbols, stories, and collective learning. All of this emerged from ancestors that, 6 million years ago, would have appeared biologically unremarkable beside countless other primates moving through the same landscapes. No observer standing there could have predicted immune systems capable of molecular memory, brains contemplating their own origins, or cultures reshaping entire planetary ecosystems. The arms race did not merely preserve the human lineage. Across deep time, it assembled one of the most behaviorally complex organisms Earth has ever produced. There is a quiet discomfort hidden beneath the evolutionary story that has unfolded across these millions of years. The capacities humans most cherish, language, imagination, music, morality, affection, cooperation, may not have emerged for noble reasons at all. They arose because organisms carrying those traits survived and reproduced more successfully than organisms that lacked them. Beneath civilization and philosophy sits an older process that never cared about beauty, wisdom, or meaning. It only preserved what continued. The implication settles heavily once viewed directly. Human intelligence did not appear because evolution intended consciousness to understand the universe. Empathy did not emerge because kindness possessed inherent value. Creativity was not selected because nature admired art. Each capacity persisted because, somewhere in deep time, it improved survival within an unforgiving system of competition, disease, environmental instability, and social conflict. Yet origin and meaning are not necessarily the same thing. A sharpened stone first created for hunting can later become sculpture. The neural systems that evolve to navigate alliances, avoid deception, or attract mates eventually produced philosophy, mathematics, literature, and astronomy. The same brain, once occupied with tracking social tension around ancient fires, later learned to contemplate black holes, compose symphonies, and reconstruct the history of extinct worlds from fragments of bone and light. The tool exceeded its initial function. Language may have begun partly as social strategy, allowing individuals to coordinate alliances, exchange information, and influence reputation within increasingly complex groups. Still, those same vocal systems later carried poetry, grief, memory, and abstract thought across generations. Music may retain traces of ancient courtship displays, rhythmic demonstrations of coordination, timing, and creativity. Yet that history does not reduce the emotional reality of music itself. A capacity shaped under evolutionary pressure can eventually become something larger than the pressure that created it. The arms race also produced far more than competition alone. Cooperation became one of the most powerful adaptive strategies the human lineage ever discovered. Groups capable of trust, coordination, food sharing, and collective defense consistently outperformed groups fractured by internal instability. Empathy, fairness, and altruistic behavior likely carried survival value precisely because they strengthened social cohesion inside vulnerable human communities. Even morality may partly represent an adaptive framework that allowed large groups of unrelated individuals to function together without collapsing into constant violence. In this sense, compassion was not separate from the evolutionary struggle, it emerged within it. The deeper paradox is that natural selection unintentionally produced organisms capable of reflecting upon the process itself. For billions of years, life evolved without awareness of what evolution was doing. Predators hunted, parasites adapted, nervous systems expanded, species vanished. None understood the mechanism shaping them. Then, gradually, one lineage developed the ability to model the world symbolically, remember deep history, and ask questions about origins and meaning. The Red Queen still runs. Pathogens continue evolving. Social pressures continue reshaping behavior. Selection has not stopped. But within the human species, the process generated something unprecedented, a creature capable of examining the ancient race that assembled it, tracing the scars left by that struggle, and turning those scars into science, philosophy, memory, and art. The war produced an observer. For almost all of Earth's history, evolution unfolded slowly. Organisms changed through mutation, selection, extinction, and inheritance across enormous stretches of time. Pathogens evolved rapidly, hosts responded gradually, and the Red Queen race continued generation after generation without pause. But somewhere within the human lineage, adaptation began shifting beyond biology alone. The species produced systems capable of changing faster than genes ever could. The modern phase of the arms race now extends into technology. Vaccines allow immune systems to recognize pathogens before natural exposure occurs. Antiviral medicines interfere directly with viral replication. Global sequencing networks identify dangerous variants within days. Artificial intelligence systems already assist researchers in analyzing protein structures, modeling outbreaks, and searching chemical databases for potential drugs. Human adaptation increasingly operates through accumulated knowledge and engineered systems rather than waiting for slow genetic change to spread through populations. Yet the opposing side continues adapting as well. Bacteria evolve resistance against antibiotics once considered revolutionary. Viruses alter their surface proteins and transmission patterns under selective pressure from immunity and medicine. The race never stabilizes. Every successful intervention creates new evolutionary conditions that pathogens begin navigating immediately. Alexander Fleming understood this even in the 1940s when he warned that careless antibiotic use would inevitably select for resistant bacteria. Decades later, resistant infections kill millions of people worldwide and continue spreading through hospitals, livestock systems, and dense urban populations. A more profound transition may now be approaching. For the first time in the history of life, a species possesses tools capable of deliberately editing its own genome. CRISPR and related technologies already allow researchers to alter specific sequences inside living cells with increasing precision. Experimental therapies attempt to repair inherited disorders, modify immune responses, and influence how cells interact with disease. Genes shaped by hundreds of millions of years of pathogen conflict may eventually become targets for intentional redesign rather than passive inheritance. The consequences remain uncertain. Evolution historically operated without foresight, testing biological changes through survival over generations. Human intervention bypasses that filtering process. A trait beneficial under one set of conditions may produce hidden vulnerabilities elsewhere. Immune systems evolved as interconnected networks shaped by countless trade-offs across deep time. Changing one component intentionally may influence many others in ways that remain invisible for decades. Meanwhile, the social arms race accelerates through entirely new environments. Human cognition evolved within small cooperative groups where reputation, alliance, and deception operated face to face. Now, those same neural systems function inside digital networks involving millions of individuals, constant information flow, and algorithmic systems designed to capture attention. Social pressure can spread globally within hours. Narratives compete continuously for emotional influence, political power, and behavioral control. Even sexual selection continues transforming. Contraception altered the relationship between reproduction and mating behavior. Cultural norms surrounding partnership shift rapidly across societies. Reproductive technologies increasingly intervene in processes once governed entirely by biology. Ancient evolutionary feedback loops now interact with economics, digital media, and technological systems unlike anything present during earlier human history. The race therefore continues, though its terrain no longer resembles the ancient environments that first shaped it. Biology, culture, computation, medicine, and information now overlap into a single adaptive system moving faster than traditional evolution ever could. The Red Queen still runs forward through the human story, but the species she shaped has begun altering the track beneath its own feet while the race is still underway. A long time ago, Lewis Carroll wrote that it takes all the running you can do to stay in the same place. When Van Valen borrowed the line to describe evolution, he was looking at extinction patterns buried in the fossil record, species appearing and disappearing while the larger struggle continued uninterrupted around them. But after tracing the path of the arms race through immune systems, pathogens, brains, social groups, attraction, language, and memory, the sentence begins to settle differently. The race is visible everywhere, once seen clearly enough. It appears in the immune cells, moving silently through the bloodstream, carrying molecular memories of infections encountered years earlier. It appears in the extraordinary diversity of immune genes inherited from ancestors who survived epidemics long forgotten by recorded history. It appears in the adaptive systems that learn to recognize pathogens evolving faster than human reproduction could keep pace with. Even the structure of the genome bears the marks of conflict, viral fragments embedded inside DNA, like fossilized traces of ancient biological invasions. The race also left its imprint on the mind. Human cognition emerged gradually inside environments where survival depended upon reading intention, maintaining alliances, recognizing deception, and navigating increasingly complex social groups. The face became expressive because social information mattered. Language expanded because communication altered reproductive success and group cohesion. Memory sharpened because remembering danger, betrayal, illness, and cooperation carried consequences measured directly in survival. Across deep time, nervous systems that handled those pressures more effectively tended to persist. The remarkable part is not merely that evolution produced intelligence, it is that a process so indifferent and mechanical generated organisms capable of examining the process itself. Natural selection possesses no foresight. Pathogens do not evolve strategically. Mutations carry no intention. The arms race unfolded through countless local interactions, repeated over immense spans of time, generation after generation, without direction or awareness. Yet from that relentless accumulation emerged a species capable of reconstructing extinct ecosystems, sequencing genomes, tracing evolutionary history, and contemplating its own origins beneath the night sky. The same process that drove invisible wars between cells and viruses also produced mathematics, philosophy, astronomy, literature, medicine, and music. A biological mechanism concerned only with survival eventually generated creatures capable of wondering whether survival alone is enough. Somewhere within the continuity of the race, evolution crossed a threshold where the products of natural selection began reflecting upon natural selection itself. And still, the race continues. Pathogens continue adapting, human societies continue changing, technologies reshape behavior faster than biological evolution ever could, social pressures alter cognition, new selective environments emerge continuously from medicine, migration, climate change, digital networks, and cultural transformation. No final stability waits at the end. Van Valen's law remains intact beneath modern civilization, exactly as it existed in ancient oceans hundreds of millions of years ago. Constant running, constant adjustment, constant adaptation. Yet there is something quietly beautiful in knowing that the thoughts moving through the mind right now, the ability to follow this history across deep time, to imagine vanished ancestors and invisible molecular struggles, are themselves products of that endless motion. The race built the observer capable of seeing the race. Tonight, while the world grows quieter and consciousness begins to drift towards sleep, the process continues beyond awareness. Cells communicate, microbes evolve, genes replicate. The ancient motion never fully stops. Rest now. The race will still be running by morning.